Kat Walsh. My name is Kat Walsh. I supervise the Ocean Standards Unit at the State Water Resources Control Board. I'll be facilitating the meeting today. With me to help with the Zoom platform is Jonathan Dolan. So if you have any issues again with Zoom, or if you would like to register, please email beach-monitoring@waterboards.ca.gov and Jonathan will get you connected. And I'm just going to roll through the attendees for the meeting, and please unmute yourself and introduce yourself if you're from one of the following agencies. East Bay Parks Department. I see Hal's on the line. Is anyone else present from East Bay Parks Department?
Kat, hold on. I just have to allow participants to unmute themselves.
Oh, okay.
There we go. And East Bay, you should be able to unmute yourself now.
Hi there, I'm Sabrina Sexton, Water Management Technician for the East Bay Regional Park District.
Thank you.
And I'm Hal McLean from the East Bay Regional Park District.
Great. And then folks from Humboldt County.
Hi, I'm Jessica Reynolds from the Humboldt County Public Health Laboratory.
Wonderful. Anyone else from Humboldt County? Okay. How about the County of Long Beach? Hearing none. Los Angeles County.
I'm Thomas Parker with LA County Sanitation Districts.
Great. Anyone else from Los Angeles County? Okay. Marin County. Okay. Anyone from Mendocino County? How about from Monterey County? How about folks from Orange County?
Hi, this is Lauren Robinson from Orange County Healthcare Agency, and I think there's a lot of us on the line, so.
I'll go next. I'm Hisham El-Mashad, Orange County Environmental Health. I work under the supervision of Lauren.
Joe Guzman with Orange County Public Health Lab.
Great.
Morning, everyone. Grant Sharp, Orange County Public Works, and also representing regulated stormwater community on the California Water Quality Monitoring Council.
Wonderful.
John Payne, County of Orange, OC Public Works.
Great. Anyone else from Orange County? All right. How about San Diego County?
This is Farnaz Farhang with the County of San Diego.
Great. And San Francisco County.
Good morning. It's Diane O'Donohue, City and County of San Francisco Public Utilities Commission.
Wonderful, thank you.
Ross Duggins, City and County of San Francisco Public Utilities Commission.
Great. And how about San Luis Obispo County? Right. San Mateo County? County of Santa Barbara?
Hi, I'm Marissa Sensulo from Santa Barbara County Public Health Department.
Great. How about the County of Santa Cruz?
Hi, it's Audrey Levine from Santa Cruz County. Good to participate in the meeting.
Hey, Audrey. And how about from Sonoma County? Okay. Any folks from Ventura County?
Good morning. This is Gladys Dobson with Ventura County Environmental Health. And I think we have Paolo joining us on the webcast, probably a couple other staff members there too.
Great, thank you. I believe we have a couple of other folks from the water boards on the line. You would like to introduce yourselves?
Hi, this is Eric Burris with the Clean Water team of OEMA, also representing the Safe to Swim Network.
Morning, Eric.
Hi, I'm Michelle Robbins. I work under Kat Walsh for the Ocean Standards Unit and will be helping with the beach monitoring program from here on out. You've probably gotten a few emails from me.
Thank you, Michelle.
Hi, I am Stephanie Webb. I'm the California Sea Grant Fellow, also working with Kat and the Ocean Standards Unit at the State Water Boards.
Hi, this is Helen Yu with the San Diego Water Board Region 9 Stormwater Management Unit.
Good morning, everybody. This is Charles Brook. I am the Microbiology Data Manager in OEMA under Tessa Fogen.
Hi, Charles. Anyone else from the water boards? All right. Anyone joining us from the Department of Public Health? Okay. Anyone joining us from the US EPA? All right. Anyone joining us from Heald Bay? Anyone else that I have missed? Great. Okay. Well, just again, a reminder that this meeting is being recorded. And anyone who's on the webcast who would like to join to speak or ask questions may submit a request to dwq-beach-monitoring@waterboards.ca.gov with the following information. And also there's, you can also send it to beach-monitoring rather than putting in dwq-beach monitoring. There's two different emails, they go to the same account. If you submit a request to register, Jonathan will get you set up and send you some information. And also please feel free to email that account if you have any issues during the meeting, or if you have any questions, you can email me at catherine.walsh@waterboards.ca.gov or this beach monitoring email. With that, I'm going to hop into some updates. Karen Black has accepted another position at the State Water Boards. And as Michelle mentioned when she introduced herself, Michelle Robbins is a member of my team and is acting as the lead staff on the beach program until we fill the vacancy. And just so you all know, I believe that the vacancy will be announced and open for applications in the next couple of weeks. So if you know of anyone who may be interested and may have qualifications suited for this position, you're more than welcome to share that vacancy with them. And again, as you've heard several times today, we have a new email for this program, and it's beach-monitoring@waterboards.ca.gov. We encourage folks to email that account for questions about the beach program and you know, things related to their grant agreements, issues with the database where they would previously reach out to Karen or myself. This is a good email account to reach out to. Multiple members of the team have access to it, so hopefully you'll have a quick response. And with that, I'll open it up to folks to share anything that they want to about the beach program, how things have been going since we last met in spring. I have some questions to get us going. One of the questions that I wanted to ask everyone is, what are some challenges you have observed pertaining to diversity, equity, and inclusion when implementing the public beach safety monitoring program? And what ways have you addressed these challenges? It could be maybe signage, having it in multiple languages, making sure that the information is readily available to different communities, available to like maybe disadvantaged communities. They can easily find information about whether or not it's safe to swim.
Hello, this is Hal McLean, East Bay Parks. We've had signage in English and Spanish for a number of years, but yeah, it is a challenge to try to reach all the different folks that come out to the beaches and all their languages. We are updating our website, East Bay Park's website, and there's been a number of ideas of trying to get things in many more languages, but it is a challenge. I don't know if QR codes makes a sense with different links, but then, you know, then you're also not including people that don't have cell phones and don't have, you know, don't have cell phones that work everywhere, and some of our sites don't have cell service. So yeah, it is a challenge, and you know, we're trying to address some of it, but there is a lot that we're missing, and guidance would be great.
Was how? Anyone else?
This is Farnaz Farhang with the County of San Diego Beach and Bay Water Quality Monitoring Program. As one of those efforts, we translated our brochures into Arabic. We have it currently in Spanish, Tagalog, Vietnamese, and I believe another language. And as part of that effort, we added one more translation. And we also have added Google Translate to our website, in addition to the QR code on our signs. That I think majority of our sites do have cell access, but that's also another effort. And we also have had our signs both in English and Spanish for many years as well.
Great, thank you. Anyone else?
As Diana Donahue with San Francisco, we also have our signage with at least 4 languages.
That's great.
Yep.
Have you guys tried any different mechanisms of how to disseminate information related to you know, why we do bacteria monitoring at public beaches, why it's important, what it tells us about how if it is safe to swim or not? Or any other mechanisms aside from different languages, perhaps, to address challenges with diversity, equity, and inclusion? All right. If anyone thinks of anything throughout the open forum, please feel free to circle back to it. Another question, maybe to get us going, is like, how have you seen the program improve over the course of the past, I don't know, 5-10 years? And what kind of direction would you like to see it go to continue improving?
Hal McClaney State Parks again. Yeah, you know, it's really improved over the past 10 years. And the Northern Beach Water Group attempted to get uniform signage, and that didn't. There's so many specifications for agencies, and there's so many multi-agency jurisdictions, and they all have their own signage and wording challenges and specifications. You know, I think we need to continue to work on it, and I think that it would be great to have like a more of a uniform signage. But you know, in the meantime, having you know, the no swimmer or some sort of stoplight thing really does cross many language barriers, especially if people are familiar with driving. I don't know. I think that the signage could get better. I really think that total and fecal are not very good indicators, and really think that we spend a lot of money on that, and think that we should probably go to Enterococcus or I don't know if there's, I mean, we keep on talking about other methods, and I'm glad we got some good things on the agenda to talk about, you know, how these other methods are working. But I see marginal to minimal value of continuing with total and fecal. And I think it would be really good to potentially include more beaches with just Enterococcus, because we're talking about maybe including a couple more beaches that got really a lot more visitation since so many facilities were closed down during COVID. People just started going to the San Francisco Bay just wherever there was sand and started swimming, and a couple places have really taken off and people continue to do it. And last summer, they still didn't have all the swimming facilities and swimming pools open. And you know, if we can have it cheaper to process everything, it might be, you know, might be able to include more places and things like that.
That's a great point.
It's Fernald's again with the County of San Diego. In terms of improvements, we have increased our number of samplings that we do throughout the county significantly, especially with the South Bay, right at the border of Mexico and US. We used to sample twice a week, and technically we're only supposed to sample once a week based on the regulations, but we are sampling 6 days a week. And thanks to the Board of Supervisors who provided the funding for us to do it. And also another significant improvement, which everyone is already aware of, is moving towards the ddPCR method, which probably we will talk more about it, but it also comes with its own challenges.
So with Orange County, our program in the last 10 years, we've been able to improve upon getting the message out to the public. Just so many more methods of accessibility to them via social media. So Twitter, email, website, in addition to the physical signs, just becoming more of a presence in getting our message out to the public so that they understand who we are, what we do. And so when our posts do go up, the significance behind them.
Great.
I just wanted to chime in with what Fernaz said, and kind of echoing what Hal mentioned, the ddPCR for Enterococcus. I mean, now that just a short while ago, EPA gave us the thumbs up to roll with ddPCR for Enterococcus on a kind of trial basis, it seems. I just think we need to have a good, strong push to make sure that we get a standardized method for that developed. And once we do, everyone can kind of get on the same board, because that's going to really decrease the turnaround time. And once we can decrease the turnaround time on that, the actual assay, we could get the data out there a lot faster. So I'm significantly on board with the ddPCR. We just have to make sure that it's uniform, and we get some actual guidance for that.
Yeah, that's a good point. And to clarify, the ddPCR method was approved for beaches monitored in San Diego County. Any other improvements that you've noticed in the past 5 to 10 years, or improvements that you think the program needs in the coming years? Great. Have there been other challenges that your local agencies have faced in implementing the public beach safety program, or any success stories that you wanted to share?
So this is Diane again from San Francisco, and to build on what Al was talking about, the signage, the city of San Francisco doesn't own any of the beaches that we monitor. It's the port of San Francisco and state parks and the Golden Gate National Resort. So it's trying to be consistent with signage is very difficult for us. But I think we've been doing a pretty good job getting signs out in multiple languages. And of course, our website does describe our program more. It's not in different languages yet. I'd like to work on that, but hopefully we'll get there. Yes. And I also want to, The qPCR methods I'd love to see be pushed forward to, and get rid of total and fecals using the MF method.
This is Sharon from Orange County. Just a quick note about the Beach Watch website. In my opinion, that could use a little bit more improvement. It has come a long way, but rather the uploading process and the way the information is disseminated throughout and the uniformity of it, I think could use a little bit more improvement.
Great.
This is Hal McLean, East Bay Parks again. You know, it'd be nice. I know we've had presentations on, you know, rapid testing that, you know, you could test every beach in an hour and things like that, and daily testing. I think, you know, it would be pretty amazing to work up to something like that. It would be good if you could somehow do it with, you know, SCADA and things and staff that are on the beaches, because I'm a compliance person. We have to run to all the beaches and run to the lab, and you know, and we do have staff out on the areas, but you know, in the future, I could see, you know, here's, you know, your morning reading, here's your midday reading, here's your afternoon reading, and you know, that and modeling can really, you know, maybe make things safer for people. But I also kind of wonder, I mean, we're in the San Francisco Bay, and you just don't know all the different other pollutants as well. And it would be good to, you know, work towards something where you say, wow, you're in this area, you know, there's potentially, you know, there's these industries nearby, or these different types of activities nearby. Maybe we should be testing for oil or some other thing that, you know, could potentially be, you know, problematic to swimmers as well. It would have to be totally regional or totally specific per beach, but you know, I mean, something like that could really be helpful.
Thanks, Hal. Okay. And, you know, kind of one of the standing questions that we've had since the pandemic started, how has COVID continued to affect your beach monitoring program? Are you guys still primarily working from home and figuring out how to get out to sample? Are there challenges with accessing beaches still for monitoring? Things like that.
Hal McLean here, East Bay Parks. We've been in the office and we've got a little lab here, but we don't process the samples here. You know, we've had people that have had close contact, and we've had staffing issues on our end. Our lab has had staffing issues. We've had building entry issues with our lab and with our with our staff, and with our lake management and lake monitoring. A lot of those were closed during COVID, but Bay Beach and the saltwater beaches have been open and have been totally mobbed, and it's kind of a good thing. But you know, I think there was times when we couldn't have bathrooms open. At the beginning of the pandemic, it was like, okay, here's all these people, and we can't have our bathrooms open. And it's just a terrible idea. And amount of trash that was left. It's just amazing that, you know, humans can think that leaving trash all over the place in a beach is a good idea. And, you know, we all deal with the public, and, but it was exacerbated by hordes of people coming and all this kind of stuff. But yeah, I mean, we're still working through some things and waiting for health department guidance and things like that. But I don't know, it actually worked out fairly well so far.
Thanks for that, Hal. And I see Joseph Palmer, you have your name up or your hand up.
Yeah, thanks, Kat. Hopefully you can hear me. Sorry, I got here a bit late. I was in another meeting there and was waiting for a little while in the waiting room. But yeah, for us, the COVID pretty much has cleared up in terms of impacts on us. With the exception of at the lab, they are very busy with COVID samples. So our samples obviously aren't the highest of priorities. So we have seen a pretty big time gap between when we're dropping off samples and when we're getting results. And it's probably about 3 or 4 hours longer than it had been historically. So that's the impact for us.
Have you noticed any improvement over time, or like over the past 6 months, or is it still a continuous problem?
Yeah, I mean, I haven't noticed it improving, other than, you know, in the initial phase, trying to juggle everything up then. There was a few really extreme ones, but yeah, we used to drop off samples at about 12 or 1 o'clock and get results about 3 or 4 o'clock the following day. And now we're dropping off samples at 8:30 in the morning. So maybe 9, but 3 or 4 hours earlier, and we're getting sample results back around the same time. So,
okay.
So that extra time in the morning is not really translated to super early responses, except on the weekends, then it has. So we sample 7 days a week here in San Diego because of the cross-border pollution issues. And we're also gearing up for the ddPCR. So we're trying to tweak our logistics, which is quite a challenge because we're meant to be at the lab by 8 o'clock. And when it gets light at 6:30, you can't catch too many samples in that time period.
Yeah, that's an interesting challenge. Anyone else want to share any challenges that they've experienced during COVID? Has anyone received any questions about COVID related to beach water quality and safety to recreate at beaches? I know that was something that we talked a lot about the last time we met. Just wondering if there were any more challenges with that or any more questions that have been raised.
Diane from San Francisco. We were pretty fortunate that we were able to monitor all of our beaches during the height of COVID, except for one, Hyde Street Pier, which was closed by the National Park Service. We noticed more swimming at our beaches because the swimming pools were all closed. Right, and then the other thing, we had some access issues at Candlestick Point State Recreation Area because the very large homeless encampment was there. And we believe they may have been impacting the water quality there. We had a lot of high counts. But the good news is that the parks and the city worked together to open up essentially a parking lot or a place where they relocated all the homeless RVs and vehicles. And it's a secure spot, but it also offers services, you know, social and waste and everything else. But we still have some access issues at Candlestick Point that we're still trying to figure out. But we're good otherwise.
Good to hear. Anyone else? Are there any other questions or issues or topics that folks wanted to talk about during the open forum that hasn't come up through the questions that we've gone through?
Hi there, Kat. It's Paulo Quinto with Ventura County Environmental Health. And I just want to say that we have been having problems with our Ventura County Public Health Lab, and we are not able to perform our samples for the past 3 weeks since their ELAB certification was expired. So we haven't heard from them, and just wondering how we can resolve this. We've tried other labs, but unfortunately, the labs that we checked did not have the certifications to perform the testing that we need.
Yeah, I believe someone on my team is looking into this issue, and I don't think we've yet found really good guidance on how to move forward. And this is something that continues to be a challenge for many, is finding labs that are ELAP accredited for the right test methods for the program. And it's something that is certainly important and that we're trying to figure out. And I think we will make some headway with it in 2022, but we'll certainly be working to work with your team to find some sort of resolution, at least for getting your sampling done in the interim.
Okay, thanks, Kat.
Yeah, I'm sorry to hear about that challenge. Does anyone else have anything that they want to share that hasn't come up yet? Alternatively, in the few remaining minutes that we have for this section, if there's something that you want to share as a recommendation for future meetings, things that we should cover, I would be interested in hearing that.
This is John with OC Public Works. So I just want to share a sort of a general comment. I think this forum is mostly on the beach water quality and recreational use protection at the beach. I work mostly on the MS4 side, stormwater. So I feel like the general findings for the folks working at the beach side is that the MS4 input remains a source of beach, high beach bacteria count. That remains the case for many years, and and now, when there is a channel that's actually flowing, usually the bacteria count are higher. There has been effort to focus on the human markers, as I think there are going to be some talks about that today by Philip and by some other folks. We have had some this effort in the past. I just want to share that for Orange County, we have some TMDL driven project that for the MS4 side and for the inland embayment like Newport Bay, we have started such effort to try to not just look at the fecal indicator bacteria input from the MS4 system, but also try to figure out the sources of human markers. However, the regulatory driver for human source from the MS4 system remains unclear. So we keep having that issue. But I think that's a shared sentiment among the MS4 dischargers that, in the future, probably we should pay a little bit more attention to not just fecal indicator bacteria, but also human sources, and try to address these sources so that hopefully, together we can help improve the beach water quality in the long run. It remains challenging, but I think that's a worthwhile effort.
Right, taking more of a proactive approach rather than a reactive approach to bacteria.
Yes.
Hey, Kat, can I jump in?
Mm-hmm.
So I've got a couple things, and I apologize if somebody's already mentioned some of these, because I did come in late. One question I had was how long it takes for everybody to get their results from when they drop off the sample. So for us, we'll drop off a sample and we get them about, I'd say 30 hours later. There's about a 24 to 30 hour delay. And then with ddPCR, we get about a 7 to 10 hour delay with that.
Yeah, that's a great question, Joseph, and that hasn't come up yet. So let's take a few minutes for folks to share how long it takes to get their results.
Diane here from San Francisco. We post and take action based on preliminary values, and they come within 18 hours to 20 hours. And of course, the final results aren't until 24, and then sometimes cases with 2 days for fecal and totals or longer.
Yeah, I just wanted to add, this is Jessica Reynolds from the Humboldt County Public Health Lab, and I am in the lab where we actually do the testing, and we go out sometimes to help the environmental health people sample, but the minimum time frame we're looking at is 18 hours for that total, and that total coliform and E. coli. That's the soonest we can read it out, and we'll give a preliminary to the guys over at environmental health, but then the enterococcus portion takes a minimum of 24 hours. So that's probably why you're looking at that 24 to 30 hour mark. And then depending on how the lab's workflow goes, they may prioritize and batch and set them up either before lunch or right after lunch. So that kind of could lead to some of that delay in testing. And we've had really good communication with our people over at environmental health. Well, we'll call them up, give them the prelim, let them know what's going on. They can go out and resample or post signs or pull signs. So just from the lab standpoint, that's what we're working with.
Do you do MTF up there in Humboldt?
We do the IDEX method. We also do MTF for different types of waters, and that is our backup method. And that one can take like 48 hours to up to 5 days, depending on what media we're using.
Yeah. I presume the San Francisco one was a MTF with a 2-day turnaround.
MF, sorry, just
okay.
For totals and fecal, and then we're using the IDEX enter alert for enterococcus.
Oh, but it sometimes takes you 2 days to get a final on the.
Not for that one, I'm sorry, that one's 24. Yeah, that one was, the enter was 24.
So East Bay Parks here. We used to get results or preliminary results in about 18 to 20 hours, and then our lab upgraded their LIM system to an XLIM system. Now we're getting preliminary results in about 30 hours, and then we're on like some 5-day thing, so we don't even get final results for a week. But we're doing,
whoa, that's really for a week.
Final results, yeah. But we post on preliminary results. We're doing membrane filtration for fecal and enterococcus, and we're using ColAlert 18QT for total. And the MF, yeah, the membrane filtration results take longer, and then they give us the prelims, and then they don't. We're on a 5-day turnaround like that. So yeah, it'll take us a week to get final results. But
do you take any action on those results? Because our Our
Yeah,
process.
no, we treat preliminaries like results, like final results, but and rarely they'll change, but it never changes enough that it would change, that it would, change. The final results aren't different enough from the preliminary results that we would have a different advisory up or anything like that.
Wow.
They're usually within a few most probable number, plus or minus 5 or 10.
So this is a really interesting conversation to have, and I actually would like to pause here because we're at time for our next presentation, and maybe have this as one of the topics that we talk about a little bit more in depth, and talking about the timing of getting results, posting on preliminary results versus final results, the challenges with the different types of test methods, and then also getting into, challenges with having test methods available to you that are appropriate for the program. And maybe we'll just carve out some time in our spring meeting for a more expansive conversation about that. I think it's worth it. And then we'll just shift into our presentation with Dr. Amy Zimmer-Faust. That works with everyone. All right. Sorry to stop us early on that. Well, I'm gonna just introduce Dr. Amy Zimmer-Faust. Thank you for joining us. She's a senior scientist with SWRP and is here to present on using ddPCR and microbial community sequencing to identify human fecal contamination at the US-Mexico border.
Thanks, Kat. And just want to make sure everyone can hear me okay. And I can't share my screen. I think it's disabled.
Let's fix that for you.
Let me see if I can address that real quick. You should be able to do it now.
All right. Perfect. Awesome. All right. How does that look? Good. Okay. Thank you.
That's great.
Perfect. All right. Well, thanks for having me first off, and excited to kind of talk to you all about some work that recently wrapped up. So today I'll be describing our efforts to characterize sources of human fecal contamination at the US-Mexico border. And this was a really collaborative effort with lots of groups involved and contributing, some of those which are highlighted here on this slide. So for these efforts, we utilized kind of this molecular toolbox approach, and the performance of those different methods is really what I'll be talking about and kind of focusing on today. So this group, and we actually just heard a little bit, snippets about, kind of the long history and the challenges of impaired water quality at the US-Mexico border region. And these impairments have resulted in numerous beach closures during both the wet and dry seasons. There's several potential sources, which I'm not going to get into all of them today, with the efforts described really focusing on trying to isolate the impacts from one of those specific sources. So this study was focused on understanding the impacts from the San Antonio de los Buenos wastewater treatment plant. And this is a facility that treats roughly 25 million gallons of wastewater a day to partial primary. And then that effluent flows into a stream which terminates to this ocean outfall. And you can see the outfall in the two pictures on the right here. So this partially treated effluent is consistently discharged roughly about 6 miles south of the US-Mexico border. And there have been modeling efforts that have been completed previously that have shown that this effluent can travel from the Tijuana wastewater treatment plant across the US-Mexico border and impact beach water quality in San Diego. But due to logistical challenges with sampling in this region and across the border, these modeling results have not been confirmed or validated with real data. So we know that understanding from potential human sources like this Tijuana wastewater treatment plant in the near shores is really complicated, and that's the case for a few reasons. First, there's dispersion and complicated mixing that can impact transport, and this can be really variable and highly dependent on ocean conditions. There could also be multiple types of sources contributing, including animal or human, to things like elevated FIV levels. And there can also be multiple human sources contributing. So really within this kind of complicated context, how do we isolate impacts from a specific human source? So the cross-border case I'm talking about today is an example, but we know that these types of challenges really are not unique to only this system. So to overcome these challenges in this study, a few different strategies were employed. First, sampling was conducted targeting specific conditions that would help isolate impacts from the Tijuana wastewater treatment plant. Second, we utilized multiple tools to look for impacts from that wastewater treatment plant. You heard a lot about ddPCR-based methods at previous beach water quality workgroup meetings. So here we use that same digital PCR-based technology to quantify 2 human markers, and we were looking for a gradient in those human marker levels emanating up coast from this wastewater treatment plant. And then lastly, we also used a microbial community sequencing technology to try and track the microbial signature of that Tijuana wastewater treatment plant in the near shore. So as I mentioned, we had this really targeted sampling to try to isolate impacts from that specific wastewater treatment plant. So this included sampling from just south of the facility along an approximately 14-mile stretch of coastline, shown here on the left, to the north, and this sampling spanned the US-Mexico border. So sampling was conducted during south swell events, so when waves were coming from the south headed to the north, and 4 different south swells were targeted over 2 different summers, with 2 to 3 consecutive days sampled per event. So just to reiterate, this was all dry weather sampling, targeting just those conditions when there was expected transport to the north, again, to try to isolate impacts from that wastewater treatment plant. So in addition, there are also questions about current monitoring, which occurs in ankle deep water, and how appropriate that is for folks lining up in the surf zone when you have this kind of impact to water quality. So for 2 out of 4 of our sampling events, we collected paired water samples at ankle depth and in the surf zone at the site sampled north of the US-Mexico border. So shown here, the map on the left again. So I've alluded to, and now I'm going to give you a little bit more detail on the methods employed. First, we used 2 different molecular methods. That digital or ddPCR was used for detection and quantification of specific DNA-based targets. Then we also conducted microbial community sequencing to look at the whole microbial community within the water samples collected. And then lastly, for comparison, we measured Enterococcus by traditional culture-based methods. So this included IDEX and membrane filtration. So what types of data did we get from each of these methods that we employed? Well, for the digital PCR, this was quantification of DNA-based human-associated markers. So we measured HF183, which you're probably most familiar with. We also measured a secondary human marker for confirmation called LACNO3. And then also by digital PCR, we measured Enterococcus gene copies. And then by sequencing, we were able to look at the entire microbial community associated with this set of samples. So this gave us information on the relative abundance of different bacterial taxa in each sample. And then we also collected and characterized effluent from that Tijuana plant so that we could generate this microbial signature for that effluent and look for it in our water samples. So for there, we were able to get an estimated % contribution at the different sites attributed to that source. Then as I mentioned, lastly, for comparison, we measured Enterococcus by culture-based methods. So first, we were interested in how these different methods compare to each other. So here we're looking at Enterococcus by IDEX and the ddPCR. And we had a strong correlation between the two, which is not surprising based on what we've seen in side-by-side performance of these 2 methods in this region during different efforts. Next, we looked at Enterococcus versus human marker results. So here we also saw a strong correlation between HF183 human marker and Enterococcus by culture, which is on the left, and Enterococcus by digital, which is on the right. We know that FIB and human marker levels are not always correlated. So the fact that they are here suggests that the the elevated Neuroproxy levels that we saw in this region are likely a result of human sources. So we were interested in not only how the methods compared to each other, but also if we could use these methods to really isolate the impacts from that Tijuana wastewater treatment plant. So the first evidence we looked for was that upcoast gradient in human marker levels from the Tijuana wastewater treatment plant during our targeted south swell sampling when things would be transported to the north. So here we have our map of sites sampled on the left, and on the right, this is a spatial bar plot where we have HF183 human marker levels on the y-axis, and sites are in order from south to north on the x-axis. This cartoon wastewater treatment plant indicates where that Tijuana discharge was. And then each plot represents a different day, days 1 through 3, that were sampled, and this is just showing one of our 4 sampling events. So for this event, we saw this clear gradient in human marker levels from the wastewater treatment plant moving up coast. Those are the red bars. And the dilution that we actually observed in the human marker levels was really similar to what has been estimated from previous modeling efforts. And then, as I mentioned, we also utilize this microbial sequencing-based approach to look for the microbial signature of that Tijuana wastewater treatment plant in the nearshore environment as another line of evidence. So here we have the sequencing results for that same event I just showed the human marker levels for. Again, this is a spatial bar plot with sites ordered south to north along the x-axis. And here each panel left to right represents day 1 through 3 of that same sampling event I just showed. So on the bottom, we have pink and gray stacked bar plot, and these are showing the top bacterial taxa associated with the Tijuana wastewater treatment plant. And we see this clear gradient moving up coast, which really mirrored the gradient we saw in human marker levels. And on the top, we have bar plots where we have the green and ground bars, and those represent the top taxa associated with the ocean microbial communities. Here we see the inverse relationship, which is what we would expect. So we also looked at how these sequencing results in terms of percent contribution that they estimated from that Tijuana wastewater treatment plant compared to our human marker results at the different sites. We saw a strong correlation with both human markers that we measured. So this is HF183 on the left and that secondary human marker LACNO3 on the right. So the sequencing results really provided another line of evidence that the wastewater treatment plant is contributing to both elevated FIB levels and the human marker detections occurring in San Diego under these specific conditions targeted. So lastly, what about how the water quality compared in those near shore versus surf zone samples, since this was another question we wanted to answer. And a reminder that these paired water samples were taken at ankle depth and in the surf zone at the site sampled north of the US-Mexico border for 2 out of our 4 sampling events. So not shown, but we didn't see any significant difference between the concentrations we measured for either of the human markers or for Neuroproxy by digital or culture-based methods between the samples taken in the near shore versus the surf zone. And the paired samples collected were also strongly correlated for Neuroproxy by digital and culture. That's the left 2 panels. And for the 2 human markers we measured as well, the right 2 panels. So the current beach monitoring at ankle depth appears to be capturing water quality in the surf zone, at least under the conditions targeted in this study. That there's some mixing going on, or enough mixing going on, I should say. So kind of takeaways from these efforts. First, really taking samples in this really targeted fashion and targeting conditions allowed us to better understand impacts from a specific source, in this case, that Tijuana wastewater treatment plant. In addition, both molecular methods utilized were useful and provided similar information about the transport of this human source in surface waters, providing multiple lines of evidence. And then last but not least, when we compared samples collected in the near shore to the surf zone, they had similar results suggesting that the current sampling is able to effectively represent water quality conditions in the surf zone kind of during these house fall conditions. So again, thanks for having me. And I think with that, I have a few minutes for questions. Joseph, I think you have your hand up. I don't see anybody else, so.
Yeah, thanks, Amy. That was excellent. I know we've talked about it before, but doesn't really get old. The one that really struck me is you had a slide up which showed the human markers versus ddPCR and also against Enterococcus. And I was struck by how closely aligned the ddPCR is to the human markers in contrast to the more spaced out culture methods. That was an interesting slide. That really struck me as being hopeful for us down here, that when we do get ddPCR, hopefully we're going to concentrate on what is a human situation rather than potentially from seals or I'm just throwing that out there. But yeah, that was really cool. And I know from speaking, I think it was John Griffith was saying that it's atypical for the ankle deep water to be the same as the surf zone.
Oh,
so
Lord.
it's slightly unusual. Is that right?
Well, let's be serious, Rich.
Okay. So 2 points. Yeah. So the Enterococcus, we were really struck by that as well. That was a pretty spectacular correlation that we saw. One thing and why, kind of one hypothesis why we think that the correlation with the culturable Enterococcus might have been not as strong is if the source of that FIB is primarily that wastewater treatment plant. It is partial primary, so there's settling tanks, and sometimes there's some chlorination that they do on the back end. So it could be that the Enterococcus cells are there but injured, and so they're not, so the culture-based results aren't kind of able to capture the Enterococcus in the way that the digital is, and that's why those two are really, really closely linked for kind of this situation where we think it was coming from this source. So that's kind of our working hypothesis for that slide. And then, your second point about the ankle depth versus nearshore, we were surprised, or I should say surf zone versus nearshore. We were initially surprised about that as well, because SCWRP has done some work a number of years ago showing kind of the inverse of that, where those two were different. That study was targeting those locations where you have like really close nearby freshwater discharges, where this one was, up coast a ways, miles from a specific source. And when we talked to like Falk Fedderson's group about those results at Scripps, who's done a lot of this nearshore modeling, and he felt like as that source from the wastewater treatment plant is transported to the north, there's so much mixing occurring that by the time we get to San Diego, it wasn't surprising to him at all that we kind of had similar concentrations. So it's promising in terms of how we do our sampling and how it would be more difficult if we did have to target the surf zone. But I want to caution our results by saying that was the case for this situation, and it could be different, as we've seen, when you kind of have different sources impacting beach water quality. So hopefully answered those. And
thank you very much.
All right, and then Eric Dubinsky, I think you had your hand up next.
Thanks, Amy. That was really great. Quick question about how do you know how approximately from maybe the modeling how long the transport time is when you have that northward flow from the treatment plant to your northernmost sites?
Yeah, so those models have shown that it can be as quick as like 12 to 18 hours when you have these kind of ripping south currents. So that was a lot faster than we initially thought. And if folks are interested, I can certainly send out kind of those modeling papers after this too, that have shown, that 12 to 18 transport under kind of the most optimal conditions for transport.
Okay, that's faster than I was expecting, because I had a follow-up question, although I'm not sure, with that short time it would apply so much, which is if you can tell the last question was about that the better correlation between the molecular methods and the culturable versus the molecular marker. And I'm wondering if that relationship is better for the culturable versus molecular, the closer you are to the source, and as it sort of, as the bacteria are in the ocean water, perhaps that breaks down more. And I was wondering if you could tell that from, maybe looking at the closer in sites versus the farther sites, but it sounds like maybe not because it's such a quick transport.
Quick. Yeah, we did look at that. And so we did a lot of these same analyses, kind of excluding the sites that were closest to the wastewater treatment plant just to see. So, and we did see still really strong correlations and pretty similar results to what we got. So we were curious about that too, if kind of the sites located adjacent to the wastewater treatment plant could have been driving what we saw, and that wasn't the case, which gave us kind of more confidence in what we found.
Thanks.
Yeah. All right, Charles, I see another hand up.
I just have two quick technical questions. Great talk. Thank you very much. One was, which reference database were you using for the 16S, or did you make a custom one? And then how are you dealing with rain in your ddPCR? How are you discriminating positive, negative droplets? Are you using the manual method, or are you using this specific software?
So I need to look back at the database. I apologize. We tried out a few, and it is, and these were published a little bit ago, but I can certainly email and follow up with you because that's a great question. As for rain, we were using a manual method.
Okay.
So yeah, we were just looking for a break in the rain.
Right on. Okay, yeah, I've been, anytime anyone gives a presentation, I always ask them because I'm trying to find some sort of standard across the board that everyone can jump on. But yeah, I imagine the reference database is Silva, just because it's such a good database for human microbiome stuff, but who knows?
We did use Silva, and then we did a second, I'm just trying to remember, we used Silva and then we used another one, and I can't remember which one we actually ended up kind of publishing our results with, because we tried out a few. That's what I'll follow up with you about.
Is it published, and where?
It is, yep. This is published pretty recently in Frontiers in Microbiology.
Okay.
So I can I will pop that as soon as I'm off sharing. I will pop the reference in the chat box.
Perfect, thank you.
Yep. All right, Phil. You have your hand up.
Yes. Hey, Amy, great presentation. Thanks for sharing all that information. It was really wonderful. I just had a kind of, I'm very curious about all this stuff, and I had a very quick question about, did you all think about doing maybe like a pseudo sensitivity analysis of analyzing when it was a northern current rather than the southern current to see that maybe the opposite holds true as well?
Yeah, so that's a great question. And I didn't show it here. I just kind of highlighted one of our events, but we actually missed the South Swell on one of our events just because it's hard to get out there and, and it's logistically challenged to kind of get everything in gear and always select the right days. So that ended up working to our benefit because we had exactly what you're talking about, kind of that negative control when it wasn't a South Swell, and we didn't see these gradients at all. And I believe we did not detect human marker during those. There was like 2 days that were kind of our negative control north of the US-Mexico border. So we did still detect it, kind of bracketing the wastewater treatment plant, but it didn't have this up coast gradient.
Very cool, thank you.
Yeah.
All right, thank you, Amy.
Thank you, guys.
This is actually perfect timing. We're going to move on to the next presenter. So I'm going to introduce Dr. Philip Gettalunga, and he's with CSU Fullerton Department of Public Health, and he's going to be presenting on HF 183 from variable sources, development and validation of HF 183, PMA qPCR method for environmental monitoring applications. Thanks for joining us.
All right, thank you for having me. I'm excited to be here to share some of the work that we've been doing at Fullerton in collaboration with our partners in South Orange County Wastewater Authority. So let me get my screen set up here and we will be right on our way. All right. Okay, so are we all looking at the right screen here? When I have these dual monitors set up, I sometimes display the notes version, but I think this is the right one here with the full screen. I'm going to be talking to you all today about our work with HF 183. We were developing a method to incorporate this differentiating ability to separate live and dead sources. And one of the kind of drivers for this study was just to make sure that when we detect HF 183 in our environmental waters, that it is hopefully more relevant to public health when we're detecting positives associated with live cells versus positives associated with dead cells. So that was one of the main drivers of this particular project that we undertook in the last couple of years. And so today's presentation is going to cover some of our main objectives. I'll talk to you a little bit about the background. It sounds like everyone's well-versed with HF 183, so I'll probably move over that section a little bit more quickly. I'll describe the viability differentiating assay, how we kind of optimized it, and how we are going to move forward and apply it in different sample types. And then we'll talk about the samples that we looked at. We looked at different types of environmental waters, as well as waters that were collected from wastewater treatment facilities in order to identify how HF 183 is starting to fractionate in terms of signals coming from live and dead cells through wastewater treatment, and then maybe what that means for wastewater effluent discharges and the water bodies that receive that wastewater effluent. So these are our main study objectives here. We wanted to modify a validated microbial source tracking method to differentiate positive signals coming from live and dead cells. We were targeting the EPA's method 1696 for the HF 183 back R287 gene sequence. And we were hopefully just going to supplement that method with a viability assay so that we can kind of differentiate live and dead signals. When we started, that was the idea, and it turned to be a lot more complicated than we had hoped. And so we ended up making a lot of changes, not to the HF 183 EPA's 1696 method itself, but how we were to kind of bridge the two assays, the two methods. Once we've worked that out, we were able to then say, okay, well, how do we move forward, and how do we understand the removal of the live and dead signals through wastewater treatment processes, because we wanted to see how a potential source of HF 183 in the environment shaped the differentiating of live and dead cells through that treatment process before discharge into the environment. And then we were going to determine if DNA from live or dead cells were actually responsible for positive HF 183 signals that were found in our environmental waters. This idea that maybe our treatment processes, disinfection processes are effective at removing the pathogens that are indicated by HF 183 is present, and maybe that the DNA, that if it does persist through the treatment process, may have a false signal associated with it in terms of how well it indicates a public health risk. And we wanted to establish some evidence related to some REC 1 waters during wet and dry periods and compare that to some of the conventional methods for culturing methods for fecal indicator bacteria. This last point here is been on hold for a while, so it's still currently work in progress. So let's talk a little bit about the microbial source tracking. And again, I recognize that many of you are familiar with the HF183 marker. Bacteroides is basically the group of organisms that represents the target of the HF183 target. It is gram-negative and it is an obligate anaerobic bacteria. And I bring this up only because when we imagine this organism that indicates human fecal contamination has been discharged from a wastewater treatment facility into the environment, this would be an aerobic environment, which may have implications on the survivability, of course, of this anaerobic bacterium, but also on the viability of this anaerobic bacterium. Bacteroides makes up 0.5% of the gut microbiome, so it is definitely an excellent candidate. And all this has been vetted out through the EPA's work with method 1696. And we have that DNA marker, HF183 BACR287, that we can quantify through a number of different PCR-based methods. We chose to stick with quantitative PCR, but obviously these are, there's a lot of work in this area of moving this towards digital droplet PCR and other technologies. It is currently the best available human-specific fecal marker. There's a decade's worth of information on the HF183 target in some form or another. And there's limited cross-reactivity with other mammals. So it's a really great method to kind of pair with our viability differentiating assay. And we're not the first, we're not the first to pair this viability differentiation with HF183, but we think we've come up with a strategy that makes it unique and makes the information that we're going to present strong indicators of actual HF183 concentrations that we need to be worried about in our environmental waters. So I wanted to show you this slide here to kind of calibrate the way that we view the rest of our results. When we differentiate between live and dead cells, the way we are presenting our data is with and without the differentiating dye. That differentiating dye is PMA. It's called Propidium monoazide. This is a very well-studied photoreactive dye that is able to bind to DNA. And so when it binds to DNA, that DNA is presumed to be coming from a dead cell, a cell with the membrane has been compromised. And so that bound DNA, when the PMA binds to that DNA, it actually will eliminate that DNA from qPCR quantification. So when we see our results, we have results with PMA and results without PMA. And most of the time, what you would expect is when you add PMA to the sample, our results would reflect a lower concentration because we are only quantifying the signals from presumably live cells. In the PMA minus condition, this is quantifying targets from both live, dead, and extracellular DNA that might be in that environmental water source. So you would more likely have a very high concentration compared to the PMA plus condition. Now, if we had a sample where the PMA plus was very close to the PMA minus condition, this kind of suggests that live bacteria are present in that sample because there would be a very small contribution to that qPCR result from dead or extracellular DNA. You can see here that the gap between the two columns here is the concentration of dead cells and extracellular DNA that are contributing to that HF183 signal. And so we use these molecular methods because number one, we're now able to start to differentiate between live and dead cells. Number two, it's a simple complementary assay to the currently accepted method. Again, PMA plus is the live bacterium only, represented by the gray columns, which is hopefully consistent through the rest of this presentation. PMA minus is the live cells, the dead cells, and extracellular DNA contributing to that signal. And some of this was driven by the regulatory environment surrounding HF183. Can we use HF183 as a guidance and as a strong input in order to create policy and create identify impacted water bodies? And so consideration of HF183 with risk-based thresholds was one of the main drivers here. And some of the numbers that have been considered in terms of what are acceptable levels of HF183 are presented up here, around 60 copies per 100 mils or 240 copies per 100 mils, depending on the ultimate source, depending on the water sample being analyzed. And so what we wanted to do was say, well, if these are the thresholds, how likely are we receiving HF183 coming from dead cells, and are those contributing to these thresholds, and is that something that we are okay with? Another driver was this idea that recycled water may be a potential source of HF183 into the environment, because if HF183 is persisting throughout treatment, then that might be a source that results in some kind of false positive result, assuming that treatment was successful. And HF183 and tertiary effluents of these municipal wastewater treatment facilities could also contribute to some of the environmental DNA that would be discharged into our local, our coastal areas, and that would have an impact on using HF183. So we are going to modify the EPA's method 1696 for HF183 BECR 287. Currently, the way it stands is we are not able to identify or differentiate between live and dead cells with method 1696. Essentially, and this is a very simplified version of the method, a water sample is collected, processed, and DNA is extracted. And once we run our qPCR amplification, we can get a quantity of HF183 in that volume of water, but we're not able to determine if they came from live, dead, or extracellular DNA targets. We just get a total number here. And the way around that is to incorporate the propidium monoazide assay. PMA is a chemical that has a high affinity for DNA. So once we have our sample that is made up of dead cells, live cells, and also extracellular DNA that might be floating around in the water, that PMA is going to eliminate the DNA signal from extracellular targets. It's going to be able to permeate through cell membranes and bind to that DNA in a bacterial cell with a compromised cell membrane. And then it will leave alone the DNA in cells with intact cell membranes. And once you've inactivated the PMA, you can extract the DNA, and the only positive signals from your qPCR results would be from DNA collected from intact cell membranes. So this is how we define the differences between live and dead. It requires this major assumption that cells with their membranes compromised are the dead cells. And so that needs to be pointed out here as well. But the end result is that we are quantifying cells that are most likely representative of the live condition. And that brings us to the first part of our project, which was this optimization of the method. How do we kind of combine the two, the PMA method and the EPA's method 1696? I'm not going to bore you with all the iterations and all the different combinations that we've tried and we've evaluated. So I'll just kind of get to the nuts and bolts of it and the end results. We looked at two different ways to treat the sample with PMA. We looked at doing a membrane filtration method, adding the PMA directly to that membrane, and then running the PMA assay and then extracting DNA. The other strategy was to run a membrane filtration analysis, collect the biomass from the membranes into a second tube, and then do the PMA treatment in that tube. And we called that the resuspended biomass method. When we compared those two methods, the resuspended biomass method ended up being the more accurate and the more efficient form of running the PMA treatment. So we moved forward with that. Once we've identified that optimal method, we then looked to see which concentration of PMA worked best to differentiate between live and dead cells. And in those experiments, we had defined the dead condition as heat treatment in order to lyse cells and create the dead condition. And so we evaluate a wide range of PMA concentrations from 10 micromolar all the way to 500 micromolar. The published literature had values anywhere from 20 to 200 or so. And so we just kind of bracketed around that. We applied that PMA concentration to live cells as well as heat-killed cells, and we identified 100 micromolar as being the most flexible concentration that worked amongst the different sample types that we were looking at. And when we were optimizing, we optimized on pure cultures, we optimized on different types of environmental samples. But those environmental samples, I should qualify, these are wastewater treatment samples, because we feel like that might have been the more complex type of sample to run our optimization assays off of. And we used tertiary and secondary effluent for that analysis. We also optimized for incubation time. The PMA assay requires a dark incubation and a light incubation. The dark incubation is to allow the PMA to bind with all the DNA in the sample. The light incubation then inactivates the PMA that is free floating in solution. We wouldn't want to have free floating PMA in solution as we do our DNA extraction, because that would risk decreasing our quantities from live cells, as PMA may bind to the DNA from the live cells after extraction. So we inactivate with a light treatment, and so we optimize that time frame. And 10 minutes happened to be the best time for the light inactivation step. There wasn't a lot of difference amongst the different times that we applied, so we just ended up choosing 10 minutes as the balance between time and effectiveness. And then we looked at our environmental matrices. Once we've optimized the concentration, the incubation time, we can apply this in a number of different types of environmental matrices. We looked at different tertiary effluents. We looked at two outfalls, San Juan Creek and Aliso Creek here in Southern California, in Southern Orange County. And we also looked at a number of different water reclamation plants before and after chlorination to kind of, not model, but to kind of identify the patterns and trends of live cell differentiation throughout treatment. One thing I did not mention was the incorporation of these controls. Early optimization methods and experiments did not really have the need for some of these extra controls because they were very controlled systems. But once we moved into our environmental samples, it became clear that we really had to identify two additional controls, which we call a non-target DNA control and a live cell control. This non-target DNA control is controlled to make sure that PMA is actually working. And so that will tell us that the PMA assay is effective. And if we got a negative result, or if we got a result that did not differentiate between live and dead cells, that's because there was really no difference between the live and dead cells in that sample, and not because our PMA failed. We also incorporated a live cell control. So this was a control to make sure that any kind of PMA that we're adding to the processing is not going to negatively impact our live cells, so that we can be confident that the live cells that we quantify have not been negatively impacted by the sample processing steps. And so here on the right is basically the PMA assay and how we kind of incorporated it into the EPA's 1696 method. All this is pre-processing prior to EPA's method. We collect a sample, we run the membrane filtration, we scrape the biomass from the filters, transfer that to a tube, and in the tube we add our controls, the non-target DNA control, and a live cell control. We add the PMA, and then we run the PMA dark incubation and light incubation, extract the DNA from the tube, sorry, that's a typo there, and then we run our qPCR for the different targets, HF183, SCITA22, and FLEC. Now, I should mention that our non-target DNA control, we used SCITA22. And if you're familiar with method 1696, this is used as the sample processing control in 1696. So this was basically done here for proof of concept to show that we can use a non-target DNA control effectively in our assay. But right now, as it stands, it's not completely compatible with the current EPA 1696 because of the overlapping controls. FLEC is a gene sequence that is specific to a pathogenic strain of E. coli that we don't typically find in different samples. And so we use that as our live cell control. For our environmental monitoring, we looked at a number of different locations. We evaluated Aliso Creek, and this is at the mouth of the creek as it emptied into the Pacific Ocean. That was at ACM1 location. We looked at San Juan Creek in Doheny Beach. We actually sampled this little offshoot here that had historical incidence of elevated HF183 concentrations. So we wanted to pick a target. We wanted to pick a location that may have a HF183 signal already associated with it, because we knew that we may just have negative results. So this gave us the best shot of having HF183 in our sample. We also looked at different water reclamation plants throughout Orange County, South Orange County, I should say. We looked at secondary effluent and tertiary effluent, which would represent the pre and post chlorination samples. And then we looked at tertiary effluent from additional water reclamation plants that had different forms of treatment goals to see if that had an influence here. At Aliso Creek, we had a number of different sampling events that occurred from October of 2020 through June of 2021. And what we found for the most part is that Aliso Creek samples were well below our limit of quantification. We had a lot of non-detects as well, and these are samples that just did not have any amplification associated with them. And these single asterisks that represent the below our lower limit of quantitation, we had some amplification, but these are not valid since they were not within our quantitative range. And so we can see here that we only had one sample that was positive for HF183. And I'm getting ahead of myself a little bit because I want to talk to you all first about when we read these slides, we're going to want to look at the controls. Our controls here, we have our non-target DNA control, and it's a very high % reduction here, which is what we want. We want to see that our mtDNA control is able to be successfully eliminated or at least significantly reduced in terms of the signal when we process a sample and run a sample. Here we saw a 98% reduction in signal, almost 99. And in our live cell control, we had about 13.5% signal reduction, meaning our live cells were quantified at slightly lower concentrations than after PMA treatment compared to samples that did not have PMA added to it. And these were considered to be acceptable levels. So overall, Aliso Creek had very low counts for HF183. Most of it was below our limit of quantification, and we had a lot of non-detects. And only during this rain event here on December 29th did we have elevated concentrations that were significantly high. So we had 261 copies per 100 mils on that rain event day. These results also suggest that dead cells or environmental DNA are responsible for some of these results, because here in our PMA minus condition, we see that this is the only one that was quantifiable. Once we added PMA and eliminated dead cells or extracellular DNA from the water sample, these counts were very low or non-detect. In San Juan Creek, I'll bring your attention over here to the controls again. We had over 90% of our signal from our non-target DNA control was reduced in our San Juan Creek sample. And our live cell control was slightly higher, but still only around 21% of the signal was reduced, comparing PMA plus and PMA minus conditions. When we look at the actual results here, we see that most of the HF183 signals were also at or below our lower limit of quantitation, which is represented by those two asterisks, and single asterisks are representing the non-detect samples. Again, dead cells or environmental DNA were responsible for most of these detections. Here you see that our orange bars are much higher than our gray bars. And so that represents a condition that would suggest most of that positive signals associated with the dead fraction. Here I would also point your attention to the October 21 sample, where we did have amplification in the PMA plus condition. So this was a little bit of an anomaly. However, the PMA minus condition, it was below our lower limit of quantitation, but it wasn't below the 152 copies by much. And when we looked at the statistics, they were not statistically significantly different. So this led us to believe that these were pretty equivalent in terms of PMA plus and PMA minus. This suggests that most of the cells, most of the positive signal from October 21 at San Juan Creek were from live cells. The rest of the sampling events that took place over the course of this project suggest that dead cells were responsible for most of the positive signals. So here on the 29th, again, a rain event within the last 72 hours. We saw higher concentrations, and this result was not statistically significant different, but it was trending towards being different. And then we have this sample here on March 12th that shows that most of the samples, most of the signal was also from dead cells at this location on March 12th. So looking at the influence of wastewater treatment on live dead differentiation of HF183, we looked at secondary effluents and tertiary effluents. Our controls, we had about 90% reduction of our dead cell control or a non-target DNA control. And our live cell control started to creep up more towards 32%. Some of the hypotheses that are were considered surrounding this increasing impact on live cells could be that some of the matrix effects of the secondary effluent and the tertiary effluent are having impacts to the cell viability, and that might be reasons why we are starting to impact our live cell control. But we continued to look at the different water reclamation facilities in South Orange and monitor the differentiation of live and dead cells throughout the secondary and tertiary treatment. What we found is that chlorination obviously is going to have a huge impact here. And so every location that we looked at, we had a significant reduction in copy numbers from the pre-chlorination to post-chlorination. So from secondary effluent to tertiary effluent. And looking at just the PMA minus condition, this is just what's happening when we're not trying to differentiate the cells. We're seeing every location has a decreased signal when we get to tertiary treatment. When we add the PMA assay, we can see that we are differentiating between live and dead cells when at these different locations throughout the treatment facility. I should also point out that the tertiary effluent here at this water reclamation plant number 2 that we analyzed was the only one that was negative for both PMA plus and PMA minus conditions for HF183 and the tertiary effluent. So that kind of begs the question, what is this plant doing differently that is able to eliminate HF183 in its tertiary effluent? This particular plant also treats, has an industrial waste stream associated with it. So we think that has something to do with why those counts are so low. Our secondary effluent from the 3rd water reclamation plant that we analyzed had a failure in the ntDNA control. And one of the reasons why we believe there's no difference between the PMA plus and PMA minus condition is that our control failed. And so we're working around how do we address a situation like this. This particular plant, the water sample was there were a lot of solids still in the secondary sample. So we believe that there was a lot of interference associated with the sample itself. The bottom line, though, is that what we were finding is that a lot of our plants that do tertiary treatment, the HF183 signal is showing up, and it is showing up at fairly high concentrations as it's leaving that plant. Of course, it's going to be diluted once it makes its way into the environment, but this is a potential source of environmental DNA for HF183. So we decided to look more closely at different plants that have tertiary treatment. And we looked at a number of different locations for that tertiary effluent in another sampling event. Again, we were seeing some of the same trends here. We have our controls showing that our ntDNA control is effective. Our live cell control is somewhat elevated. But we saw, again, some of the similar trends that in all of our tertiary effluents, aside from water reclamation plant number 2, which again is the one with the industrial stream component to it, we saw concentrations of HF183 in that tertiary effluent. And some of them were very high. We had one of our plants had extremely high numbers compared to the rest of our facilities here. So HF183 is present in the effluent. The DNA is absolutely able to persist beyond tertiary treatment in some of these locations. We are aware that other treatment facilities have been negative for HF183 in the tertiary effluent. So we're very interested to continue the work to understand why we're seeing some of these differences between our sample locations and others. We understand also that the dead cells from the disinfected tertiary effluent are a potential source of environmental HF183 signals in our environment. We see here the concentrations per 100 mils are posted here. Again, water reclamation plant 4 is extremely high. And we don't quite know exactly what this means in terms of how it influences our environmental monitoring, but I think it begs some attention here. And then when we look at what PMA treatment is doing throughout the sample set here, it has significantly reduced the concentration of the HF183 targets in tertiary effluent to below our lower limit of quantification. We did not have any non-detects, so we still had some amplification, but again, that was well below our lower limit of quantification, and you would be considered out of our quantitative range. So PMA treatment is able to remove that signal significantly from all the water samples that we analyzed. And just to kind of sum it up, we wanted to say that DNA from presumably dead bacteria are largely responsible for some of the HF183 detections that we were finding at Aliso Creek and San Juan Creek to some extent. There were periods of time in October that San Juan Creek showed mostly no difference between PMA plus and PMA minus conditions. So that would have been an event that we would have paid a lot more closer attention to. But for the other dates, the PMA plus condition had significantly reduced that signal. So we would say that that would potentially indicate a lower risk to the public. HF183 concentrations in recycled water is influenced by DNA from live and dead sources. And this is a concern that we have is if we continue to use this marker without this ability to differentiate live and dead cells, are we sure that the concentrations that we're finding and the standards that we are thinking about are actually relevant to our goals of being protective of the public, so of the public health. So something we really want to consider here is potentially adding a viability differentiating assay so that we can get a more accurate count of HF183 in terms of how it represents the risks to public health. But again, we do have some, a whole host of limitations associated with PMA. PMA needs to be validated for each target microorganism. So we had gone through this lengthy validation process using PMA on Bacteroides. And Bacteroides, worked well with PMA, but other targets may not work as well. So if we wanted to adapt this method to other indicator bacteria or other targets, we would want to validate it first before applying it. The effectiveness is limited to microorganisms with compromised cell membranes. So when we talk about how cells may be inactivated by UV, that has a different implication because we may not be able to effectively differentiate cells inactivated by UV compared to cells that have compromised membranes. And then this whole additional assay of adding a PMA differentiating component to a method is going to add additional processing, additional labor, and also be a potential source for sampling loss or mishandling or lower rates of recovery. So we are aware of those kinds of limitations. But with that, that is the conclusion of our study. I want to thank all of my collaborators at South Orange County Wastewater Authority and the SAQA Project Committee 12, our Science Advisory Board that was instrumental in putting together our experimental design and having fruitful discussions about the data were very important in this project. And my students and staff that helped along the way as well. I'll take any questions at this point. I'm happy to answer anything today, or if you have questions, you can also email me as well. Yes, I see a hand. Alexander, I see your hand up.
Yeah, hi. Thank you. Super interesting talk, and we work with Orange County on the method too. And I have one question. So you mentioned the EPA methods. Have you tested what the differences between running your PMA treatment before the method and without?
Yeah, so you mean with an unmodified EPA method 1696? Is that
Right,
what you're
exactly
asking?
right. So if you can compare it, or if you have to run as a third sample with the EPA method by itself to really have the method.
Yeah, so we've looked at earlier on. We looked at a direct comparison with EPA's method 1696, and it really was elevated in concentration. I didn't show any of the results here, but it did have higher concentrations compared to with PMA treatment. But we were using a cyber green assay, and rather than their TACMAN chemistry, but we were still using all the same primers and all the same processing, but it was a slightly different assay. But it was a higher concentration than once we added PMA to the assay.
Okay, so even compared no PMA with compared to just the EPA methods.
Yes.
Right? So you saw higher. Okay, that makes sense.
Yes, Eric. Yes.
I'm wondering if in any of your studies you also looked at just like the culturable indicator bacteria, such as the Enterococcus idax test, to see how that is it, because you would expect that would correlate better with your live fraction.
Yeah,
I think. And kind of following on Amy's talk, she saw that better correlation between the two molecular methods than between the culturable and the molecular HF183 marker. And so, that's potentially an explanation.
Yes.
And so this would help resolve that.
Yeah, absolutely. It's a great question. We are aware that would have been a nice addition to a lot of the work that we have done. And it was actually part of our next task, is to incorporate comparisons directly with culturable and the PMA assay. But we haven't made a lot of progress on that end. We are still trying to move forward with that. And we will be looking at Enterococcus specifically. Yeah.
And has anyone done this, or do you have any plans to do this with the molecular Enterococcus test? That would also be useful.
I'm not aware of the PMA assay being modified or added to the molecular Enterococcus test, but it wouldn't surprise me if it's in the works somewhere. And we did not have immediate plans to do so at this time. But that's a great, we would definitely, I'm sure Sakwa and others would be very interested in these kinds of things.
Yeah, thanks. Great work.
Yes, Helen, I see your hand up.
Great work. Thank you, Philip. I just wanted to ask that, could you explain again what is the role of the live cell control? Because if they are live cell, wouldn't you expect that the differences with and without the PMN treatment should be similar? So the reduction is expected to be as minimum as possible. I don't know if my understanding is correct or not.
Your understanding is correct. In theory, it would definitely be like negligible difference between the two. But in practice, what we've seen is that when we add that live cell in there, there could be environmental matrix impacts to the live cell itself. And so it could injure it during the processing. It can impact the cell membrane. Maybe there's an osmotic balance issue. And so when we add the live cell plus the PMA, it now is able to infiltrate our live cell and cause that number reduction. So that's what we were seeing with our as our sample types change, we saw differences in the live cell control go from 10% up to 30%. And so, yeah, you're right. It shouldn't be that high, but we think that it's caused by, the environmental matrix effects from the sample itself on our target cell. We would like to resolve that, and maybe we can think of a way to have a more representative sample. And the idea behind that live cell control is that we can measure the impacts of the PMA addition, but what we didn't anticipate initially was the impacts of the environmental matrix. So we're still having to resolve those differences there.
Thank you.
You're welcome. Alexander, yes.
So I forget another question. So you mentioned potential matrix effects, especially like solids. So have you done a test trial with different solid contents to see if the solids absorb the PMA, anything like that?
Yeah, so we haven't done a formal like turbidity analysis or suspended solids analysis on some of our samples. We have some of that data, so we're still kind of in the works of analyzing some of that and figuring out if they impacted our quantities and if there was a relationship there. So we haven't finished looking at all that data, but we definitely are interested in the role that material has on our PMA quantitation. We're also interested in how the chlorination process can impact tertiary effluent samples in terms of the HF-183 signal. So we're looking at chlorine content and all these other parameters to determine their impacts there.
Well, thank you so much for your time and for presenting, Phil. It's great to have you here. For folks who wanted to reach out to either presenters, we provided their contact information on the agenda, so you can follow up with additional questions if you have any. At this time, I'm going to prep us to move on to our next presenter. This next hour of the meeting is primarily for recipients of the Public Beach Safety Monitoring Program grants from the State Water Board. So if you are not a grant recipient, we ask that you now take either you can listen in if you want to, but it's grant focused, it's about invoicing. So if you're not a grant recipient, you're welcome to leave at this point. With that, I'll introduce Amy Measel from our Division of Financial Assistance at the State Water Resources Control Board. She is one of the folks who helps us with invoicing and managing the grants.
Hey everybody, can you hear me okay?
Yes.
Okay, awesome. Does anyone need like a mini break to use the restroom or something? I know you guys have had a long meeting before I was able to join. No show of hands. Okay, then I am just going to begin with, I'm going to share my screen. So hold on, first you guys are going to see yourselves, so I apologize. Okay, oh, we do not want to see all this. Hold on, there we go. Sorry, I'm used to working in Teams and I've got that down. Zoom's a little different when I actually have to present. So, okay. So today we're going to be talking about disbursements and invoice training. I know there's been a bit of confusion around that, and that's why I offered my services today to kind of level set everybody and kind of start you guys all from scratch on where we are right now with what we need for invoicing. So today we're going to be going through the roles, the review process, final disbursement dates, backup you guys are going to give us, common mistakes, what PD certifications need to look like, and then the allowable signatures, because I know that's been a big one. I've given some travel resources. There's other resources, and then if anyone wants to stay on, I will demonstrate how to sign the RR. So that is kind of what we're going over today. And I'm going to provide Kat with a PDF version of this. So it'll be printable for you guys. She can send that out, or Michelle can send it out, so that each of you can have a copy of all of this. So there's a lot of words on some of my PowerPoint presentation pages, but that's really for you guys to use in a printable form later on if you need this as a reference. So your guys' roles as the project director, or anyone who is submitting invoices, because I know it's not always the PD, but the PD specifically is the primary contact for the project, and they have to make sure financial controls are maintained and that, you know, we have all the documents that we need in order to perform a review. And then you're going to certify those invoices and the progress reports and make sure everything's accurate. So that's hopefully going to reduce the disputes. But just note that when invoices are electronically signed, they do need to be sent from a designated email on file, which is on the PD cert that I know you guys have had a lot of questions on. So we will go over that in a little bit. Otherwise, you would have to send your invoices in wet, which is hard copy. And we're in an age now, because of COVID, that I think we can really move forward from having to submit wet hard copy invoices. So the project manager's role is to manage the project for us. They're your primary contact. So currently that's Michelle and Kat who are overseeing that for you guys right now. They're then going to review the invoice backup and dispute things that are necessary or ask for extra documentation if something's unclear. Then the invoice will be approved. And then they are going to ensure overall that the costs of the invoices are in alignment with the budget, are in alignment with how far the progress report has gone. And then they maintain their own technical auditable file. So the DFA has their file with your program analyst, which was Melissa and is now Cassie. And then your PM has their file, and then you guys hopefully have your own files. And then they are going to communicate with you and help you keep the project on track. So especially entering this third year of the agreements, it's going to be important that the projects are on track because they are going to be wrapping up for this session. Excuse me. Your program analyst, who is Cassie and was Melissa, they're an assistant basically to the project manager in obtaining pre-execution documents, but also if there is any time extension or amendment or budget shift that's required, that's the person who would be in charge of helping the PM deal with that. And then they have their own project closeout procedures. And then again, they have their own file. And then when it comes to my role as a DA, I make sure that you guys have your invoice workbooks and your templates. You should have all received new ones for this year. There were just several errors, and there's been some updated language because I know a lot of people had questions about the invoice workbook still said you had to sign it wet. So I went through and I updated individually all 16 projects with all of that language so that each of you has the most updated workbook possible. I also provided instructions for everyone and of course a new copy of the RR for this year. And then when I get your invoice approved from the project manager, I then prepare it and process it for accounting, who then sends it to the state controller's office. And then they issue your check, which is why it takes a little bit of time after I have told you that it's been forwarded to accounting. But those emails that I do send you as a notification are very important that you read. If there was any kind of dispute and the previous or total expenditures has been changed, or current expenditures even, that's going to affect what your next invoice looks like. And those are the kind of errors that I see more than most people. So it's going to just be important that you look at those emails that I send and the attachments, especially the disbursement tracking sheet. It's a great tracker for you guys to even just steal yourself and type in stuff, because when I give it to you, I've got my own copy. So edit the heck out of it. All right. So again, the invoice review process. The PM is going to review everything, and then they're going to forward it to the disbursement unit, and then it's going to be assigned out. The RR has a specific format that SCO will accept. There are certain things that I have to edit to make sure it matches all of that. So it is appreciated that you read the instructions that came, because adding extra information makes my job harder. Especially if it is wet signed, it's very difficult to edit the PDF when it's handwritten and it's mistakes. And so just fill out the 3 little things we'll go over in a little bit. Then I'm going to send that RR to my manager, and she is then going to approve and make sure we have lots of checks and balances here, make sure that it's in the format that's needed. And then I will then, once she signs that, I will forward it to accounting, and then I'll send you your email. Accounting will then review it, forward it to SCO, and that's when you get your payment. The final invoice for a project should be indicated on the disbursement form in this box. There's a box number 8, and it says check if final invoice. So that's going to be important for everybody's final invoice this year, because otherwise I have to have extra communication with Kat and or Michelle or whoever the PM might be at the time. And it actually says in your grant agreements in this section that you need to check that. So it's greatly appreciated that you check that little box before you sign it. And then, so when you're submitting your invoices, the cover page of the grant agreement that you guys all have and the submittal table all state really important dates. And for me, the final disbursement date is the most important. But you will see that you have the work completion date as well. And all of you guys are supposed to be submitting your invoices quarterly. I know the first year was really tough because the agreements were not executed as quickly as I think everybody hoped. So I got a lot of invoices that was just one for the whole year, and that's totally fine. But now it's going to be important, because the more of you submit invoices at the last minute in May, the harder it is on me. And I have 100 projects that I'm carrying right now. So I don't want you guys to get buried because your final dates are going to be really important. So all of you must give us your final disbursement request before June or July 31st of 2022. Okay, that's going to be really important. And that's stated multiple places in the grant agreement. Fiscal year for beaches is July 1 to June 30. They're all 3-year fundings. You can see how on the disbursement tracking sheet that I provide with every invoice, I have given each of you guys, I've divided it up so you can see what was paid out of which year. If there's any edits to that, you guys can do whatever you need to, because I have to keep my own tracker as well. And then for those of you who have not yet submitted your first invoice for Q1 of this current fiscal year, it would be the next invoice number. So if you submitted 8 invoices before, you'll start with number 9. If you submitted 4 before, then you will submit invoice number 5. So they do have to be consecutive though, even though it's just for the life of the project. And what's really important, if you think that you're not going to be able to submit your final invoice in time, you need to tell your PM immediately and before the final disbursement date, because there's a process that we have to do that in the event that this happens. Because I know sometimes our subcontractors are not very good at billing us in a timely manner, but it's very important that you do that, because there is a process that has to happen in order to make that happen. All right. Any questions so far? All righty. Okay, so when it comes to backup documentation, these are examples. It's not a complete list, but any invoices from subcontractors that would help the project manager determine where your costs came from and if they're eligible, laboratory costs, any travel information. Not required are timesheets from the subcontractors or the vouchers of what you guys have paid. So it's the check that you wrote to them. It's not necessary. We just need to see the invoice, but you do want to keep that kind of stuff in the event of an audit, because they will want to make sure that you've paid those subcontractors within 30 days of receiving the check from us. Backup does have to be saved for the life of the project specified on the cover page of the grant agreement. So in some of my projects have a 29-year life of the project. Some only have 7. So make sure you check your grant agreement because it does explain that further. And then you do want to make sure that whatever documentation you do submit is documentation that in 10 years, when you get audited and you're no longer there, are the other employees who currently work there going to be able to decipher what these documents meant for the auditor, because they're going to be going in blind. So just making sure that things are labeled clearly so that it makes it a lot easier for your future peers or the people who take over for you, as well as the auditors, because the more organized you are, the less deep that people are going to look, you know? So these are just a few examples of eligible and ineligible costs. So obviously direct project costs are going to be something that are eligible. So that's going to be those laboratory costs, any admin that you guys have, specific tasks listed directly in the grant agreement, and then travel that's in alignment with the Cal HR guidelines, which we'll go over a little bit later. Ineligible costs will include food and beverage for maybe big meetings that you guys have. Like if this meeting today were provided by one of you and you provided food and beverage and it was in person, then that kind of food and beverage would not be eligible. Any legal fees are not eligible, fines, penalties. So, I mean, it all kind of makes sense. It's the state will pay for, or the federal government, depending on the type of project, right? It will pay for things that are within reason, but if you're not paying your bills on time, we are not liable for those late fees. So common invoice mistakes, and I'm not going to go over every single one because you guys will receive a copy of this, but a couple of things I would like to point out. Missing signatures. We have to have signatures. It's very important. So please make sure that we do have that. And then if you do end up needing to resubmit an invoice, you need to put the original date of the invoice, because potentially if you do not, all the other dates will be out of order. So please make sure if you're asked to submit a new form, the original invoice date goes on the disbursement form, and you will not even need to put it on the RR because that's my job. So just the original invoice date is going to be the most important. And then another one I'd like to point out is just the line items being overspent. I know that budget shifts are very common in these projects, so please keep doing that, but try to do it with some forethought. So if you are over budget and you put that you're over budget rather than asking for a budget shift, you're going to have to resubmit the forms again. So you can save yourself a little work if you see, oh, I'm going to be overspent on this. Okay, I'm going to ask, Michelle, if I can do a budget shift, and she and Cassie will deal with that. And then I'll send you out a new workbook, and that way you only have to fill everything out one time. So it can save you some time if you just see that you're going to be going over a little bit. And the submittal summary, we are not going to go over this bullet by bullet, but a couple things I wanted to point out. If you do have something that maybe it goes under personnel services and indirect, you have one bill for everything, you put those on 2 lines and then you would divide that information up. So if you can see in blue, there's also some ways that you could potentially explain things. So like adding a very long explanation is great. That box expands, it gets bigger. So that's really wonderful. and then referencing the item number 2 there. So the whole amount was a $5,800 invoice, but we're only charging for $2,000. You can explain why here, and it makes it a little bit easier. So, and just know that when you do scroll down at the summary sheet, there are directions. So as indicated right here, I put that on everybody's sheet because I know that you guys might not be the ones filling this stuff out. It might be somebody else. So I've tried to make it as easy as possible for everybody to be able to do that. But you can read this at your leisure. Just know I did have, it's not remained. It was supposed to say remainder. So just know I have a little typo on there, but I'd already done a screenshot, so I wasn't replacing that. Any questions about the summary sheet? Because I know it's been something a lot of people have had questions on in the past. Alrighty. So your mailing address does matter. What is on the RR form, because that is what goes to the state controller's office. It's extremely important that that is what matches your project and where your check is going to be sent. So at the beginning of the project, you guys were all required to get the project or the program manager or the PA, so the program analyst, the 204. It's used for those checks. It's required for every address change if we don't already have it on file. So because you guys are counties, you all have a ton of addresses. So I think it's Mendocino, they have 61 addresses. LA County, you probably have 110. So if we don't already have the address in our financial system, I'm gonna need a new 204. But when you are telling us about an address change, you should also inform your PA and your PM because it's very important. I know some of us have, or some of you have discovered some errors in the address on the RR forms because maybe I transposed something or you recently changed. So I made sure that I CC'd both of the people that needed to be CC'd on that, but they need your contact information just as much as I do. So just make sure we're all fully aware. If you do need to fill out a new 204, I've given you the website there. It just won't be filled out with who to give it to, but when in doubt, give it to your PA and your PM if you think that that new address is not in the system. And next, we are going to look at an RR form. So it looks like a lot of information, but there's only 3 places that you guys have to look at and deal with, which is really nice because a lot of people used to handwrite all this and it saves me a lot of time when it's not handwritten. So you're going to enter your requested amount and then you're going to use the little, there's a drop down menu here for a date. And then you're just going to click to sign. And after this presentation, I can stay on so that everybody can actually get walked through how to create that signature. I also have a document that helps that, and I'm providing some websites that can help you guys with that as well. Please note that the RR form, in order to keep all of these fabulous blue areas and my formulas intact, it has to be sent as a separate attachment to your invoice package. So I do have directions on how to do that, but I mean, it's very simple. If you just, when you do have this little box that does get signed, it'll prompt you to save it, and you can just save it to your desktop or whatever file it goes into. This is the filing convention that we use here. So if you want to name it for that, or you can keep the names that you guys have, it does not matter, but it just has to be sent as a separate document from the rest of the invoice packages or package. And then I get to fill out the rest of the form. So you don't even have to worry about it. You guys just have those 3 boxes, which is really nice. I wish they had asked us for some input and made it yellow because it would have been easier for me to say, fill out the yellow part. Now I have to tell you guys to fill out the very light gray, almost indiscernible through the white, but that's okay. All right. So we're going to talk a little bit about the PD cert because I know this has been a little bit difficult. So the PD cert is essentially the document that authorizes us to talk to someone rather than the authorized representative, who is usually a appointed by the board. So that's usually someone who doesn't want to deal with the day-to-day stuff. So the PD is our day-to-day person. However, the authorized representative has to fill out the bottom of that PD cert so that we know that this one person, in this case, ABC, is authorized to talk to us and sign things, and we can talk to them. So this document must be signed with a wet signature. So you would print it out and write it out like option number 1, or it can be an image of a wet signature. So this is someone who had written their name, took a picture of it, sent it to themselves, put it into Adobe, and used the fill and sign feature. We cannot have a digitized signature like this. You cannot use the fill and sign. I'm going to put a fancy font in there. Like, you can't change it to Lucille handwriting and make it acceptable, because I have to compare your handwriting to what's on the a wet signed invoice, because those do still happen. And then you can't do the whole, I'm going to, make it look like a poor little kid did it by writing with a mouse. So, because anybody can write anybody's name with a mouse. So on a PD cert specifically, only these 2 types of signatures are allowed. And when you're filling out the PD cert, it's very easy. I'm sorry if you guys can hear the blender in the background that my husband decided to turn on right now. So the authorized representative is going to be in black. So I'm sorry. Those are the designees. So let's start with the PD. The PD is going to be in blue. So there's a name, a phone number, an email address, and then they're going to show their signature down here and the date that they signed it. Any designees, so these are people who are allowed to fill out invoices and sign for invoices, but they're not necessarily going to be any kind of negotiable power when it comes to the grant agreement. So they are going to be this black section. Now, those are the people that I really look for when it comes to, did this person send from a designated email? Because again, we're talking about these electronic signatures, has to come from one of the emails on file. So let's say though that you have, an analyst that you're going to let them send all this stuff after someone else signed it. What you could potentially do is just type in their name and type in their email, but do not allow them to sign it if you only want them to be allowed to send a package that was signed by the designee or the PD. So that is a possibility. If you do not provide a signature, they're not allowed to sign it, but they could send it. And then of course, the authorized representative that we talked about a little earlier, they would sign the bottom, because when the PA receives this, they have to see that the authorized representative is the one designating all of these people. So that's why it's important that that last section is signed. And again, these need to be an image of a wet signature or a handwritten signature or a wet signature on the PD form. And when it comes to the acceptable signatures on the disbursement form, so this is the one that you're going to list all the expenditures in your 4 different categories. You can do these first 4, okay? So you can do, this is our preferred method because this number 1 method is the only one that keeps my formulas intact, which makes this form a 3-minute process instead of a 25-minute process. So it's always going to be preferred that you utilize the click and sign method. And again, we can go over that a little bit later. You can use the wet sign. So if you do scan it and wet sign it, you can do that. You can do the actual image of a signature. So that's that one where you've kind of uploaded it, but it's your real signature. And in this case, you can do the terrible little fill and sign. I'm going to draw it with my mouse version, though, I mean, it's going to look like a 3-year-old did it. So when the package, including the R, is electronically signed, though, again, it does need to come from an email address on the PD cert. And on the RR, there's fewer signature options that are allowed. The acceptable signature that we really prefer, again, is the one that is the Adobe digital image. And just so you know, and as you guys can see on your own RRs, I actually have an image of a wet signature here, and I can show you guys how to create that. But this signature is also acceptable because you had to put in your password and it can only come from you. Or again, you can wet sign it, but that gets rid of all my favorite formulas and makes that process of filling everything out a little bit harder. Though if there is a choice of I have to wet sign everything except for one thing, the RR is the most important to sign electronically, please. Let's see. Okay, a little bit about travel. I've given you guys the travel page here because you guys have to follow the guidelines that we have to. But I'm just going to go over, like you guys can read. It's a very extensive website and it's not the most enjoyable. But just know that when you have personal use vehicles, date of travel, distance, and rate must be included when you're claiming the mileage. For fleet vehicles, you have to show the rates and the dates that the fleet vehicle was used, and your project manager will likely want to know why you used a fleet vehicle, though in some cases it might be obvious. So, you know, when in doubt, let them know, but we do need to see the rates and the dates. You can't just claim vehicles. And if there's any hotel stays and meals, you need to include the receipts because those have to be verified against what the website says currently. Okay. Any questions or comments? Because that's my presentation. And I can't see anyone raising their hands. Hold on, let me see. There we go. It looks like Joseph.
Yeah, I'm just back one page there.
Yes.
Look like we can use fleet costs. That's what we only use pretty much our fleet of trucks because we drive on the beaches with them.
Yeah.
The cost, it's got to be $1.10 or something like that per mile. So it's significantly more than the standard rate.
Yeah.
So I thought that we had gone back to not being allowed to do that anymore.
That might be something specific to beaches. As far as I know, if you're having to use a fleet vehicle, you have to use a fleet vehicle. I also do invoicing for different bonds projects, bottled water projects. Like I have everything. I've got over 100 projects that I work on. So fleet vehicles is allowed as far as I know. If there's other communication that's not allowed, what county are you from?
San Diego.
San Diego.
So we
It
don't
might
have
be
one of the 3 years. We just have that every year we have to.
Yeah, you're one of the ones that has the new project every year. I wish you guys could have another, but I think it's because of the federal funding.
I think we're hoping this is the last year of doing that, and then we're going to share the federal funding load to others.
That would be nice. It would make things a little easier on our end too. But I do know there have been other projects that have done a fleet style vehicle. So you want to do the cheapest method, but I can't drive my Sonata on a beach. Like it's not going to go anywhere. So you have to use that. So you might need to one off with Michelle and Kat and find out specifically what that is for you. So, but I'm just saying, if there's a fleet thing that you guys are using, there's specifications on what you have to provide in order to do that. And you would always want to use the cheapest method because the state does not want to pay for anything that's more expensive than you could have found it cheaper. So, you know, if an economy vehicle would suffice, that's what you're required to have. If you don't have 5 people in your car, you get to drive the little tiny, not cute car. Sorry. So, but maybe one off with Kat and Michelle on that one. They might have a little more insight because I don't usually see the backup for beaches.
By fleet, I mean not a rental, but like our own fleet of vehicles.
Yeah, like a county vehicle.
Yeah.
Absolutely. So, and each county is going to charge for that a little bit differently. I've also seen fleet vehicles for subcontractors in some of my other projects. So it doesn't happen very often, but there are some allowable things, like it needs to not be outrageous. You know, they might say, no, we're going to reduce this to the mileage per mileage of the state. They might, like Kat and Michelle might reduce the cost down to that, but at least it would be some reimbursement. So yeah, one off with them and see if there's anything they know a little bit more about that. I'm sorry, I couldn't answer better.
Okay. Thank you.
Yeah. Any other questions that anybody has? I'm not going to make a show of hands for looking at how to create an RR form, but I do have a live RR that I would like to go through. And then I have a creating of a wet signature or a likeness of a wet signature. So if anyone wants to do that, but I also provided the Adobe versions of how to do it. And I created my own document as well, because I think their version might be a little bit confusing and I like to have screenshots of a step-by-step. So I'm going to provide that to Kat after, so that can be provided to you guys. But if anyone wants to jump off, I won't be offended. But if anyone stays on, then that's what I'm going to do next, if there's no more questions. Okay. So am I still sharing my screen? I can't tell. Okay, it does say that at the top. So I have this document that I created on how to configure an Adobe ID. So I think it's only like 4 pages, but it's because I made the pictures pretty big. So I'm just going to go through and I'm going to create one for you guys, but provide this to you later. So it might be kind of fast, but it shouldn't be too complicated. So after, this is the RR template before everything has been fixed, and all of the green items are things that I have to fill out prior to sending it to you. So I would then go and sign. So I'm going to go ahead and pick this top. Nope, I'm going to pick, I don't know. I'll be a Beaches DA because I created some signatures here. And what I'm going to do is configure, or I could configure a new ID. So I'm going to do that first for you guys. I'm going to create a new digital ID, continue. I'm going to press save to a file because I wouldn't know how to delete my the Windows certificate store ones if I ever wanted to delete them. So I always save them to my computer or my OneDrive. And here I'm just going to create one called Beaches DA. And then I'm just going to be beaches at beaches.com. Whoops. Okay, I'm going to press continue. So this would obviously be your contact information. And then I'm going to save this. I don't want this saved to my pictures. I'm going to save this one to my desktop. So I'm just going to put that right there. And then I'm going to create a password. Whoops, hold on. And then you, of course, have to confirm your password. And then you press save. So now I'm going to identify my Beaches DA one. And I'm going, so that I literally just created an ID. That's how difficult it is when you push on that button. Now I'm going to actually continue. So here I have several different options. So I want to edit this because if I'm going to be the disbursement analyst, I'm not a project director. So I'm going to go ahead and edit. And this is where you can draw your little thing. You can text, and this would end up being your name, or you can select an image. So if you need to select an image, you can go to browse. So I have several different signatures in my pictures because that's where I like to keep them, but it's like, oh no, it doesn't look like I have them. So then you would just go, I know it's probably small for your guys' screens, but you go to this little drop-down menu and you can find your JPEG pictures. And so there you're going to end up with a lot of different options potentially if you have more than one saved image. So I literally, you know, drew on, I put lined paper behind and a white piece of paper on top, and I signed until I found signatures that I actually enjoyed. You can see that I did not like my signature several times. And then I have my initials there. So then I took a picture with my phone in very good lighting. I edited it so it would be as white as possible. And that's where I got signature number 3, which is the one I'm most pleased with. And so that is the signature I use now. And then I sent that to myself and then saved it to my desktop. So that's how you go about doing that signature. It's a little bit time consuming, but it's not that difficult. So I'm just going to choose these initials for now. And then you can see it changes what my image is going to look like. And I'm like, okay, this is the signature I'm going to use. So I'm going to go ahead and press save. I'm going to punch in my password, and then I'm going to press the sign button. and I'll just keep it as an example, and I'm going to override. Do not override yours, because if you're overriding it, that means you're overriding your template. And you don't want to do that. So actually create a new name for it. But now you can see that I have a signature, and you can see all of the blue boxes that were in existence where the green is they're all gone now. But I still have the ability to utilize all of these other features. So when you guys receive it, it's going to look like this. Okay, and the 3 things that you have to do are, again, you're going to enter in your dollar amount, you're going to pick the date that you're signing, and then you're actually going to sign. So this is a project director now. So I'm going to go ahead again, I'm going to edit that because you might have to click away to get to an actual image. Oh, I'm sorry, I forgot you have to browse. And then that's where you can find your, I have an image. So, okay, I'm not going to worry about it. I'm just going to browse and use one of my existing pictures that I have. Again, if you can't find them, go to your JPEGs, and then it'll change that picture. And then you can save that and then punch in your password. You can see I have a ton of different IDs, and that's specifically for this. I'm going to go through and delete everything. Looks like I can't remember the password for this one because I have so many now. Okay, well, I'm not going to sign this one because I have another example, but let's just pretend there's a signature there. So now there's a signature, okay? So don't forget your passwords, otherwise you have to configure a new ID. And then the next thing that I'm then going to do is everything that's in purple. So I'm going to take care of all of these other things. So you just have those 3. And let's say that you sign this on this date, but you put the wrong day here. I can still fix that for you too. So don't worry about all that. I can go back and I can make the change. Just note that the invoice date has to be after the DA signed it, of course, but before or on the day the project director signs it. Everything needs to be in order, because the idea that accounting has is you cannot create an invoice after you've signed it. So that's why the invoice date is going to be so important. And I pull that directly from the invoice date that you guys put on the disbursement form. And then after I have completed all of those steps, then we will have a completed RR form. So we've got the date stamp that's required and all the information that we need. This invoice has a reduction of costs, and then I sign it, or my boss signs in both places, and that's the last step for the RR. So you can see, even going through it, all of that stuff would only take me 3 minutes to do if you maintain my formulas for me. If you do not, and you sign it wet or scan it weird, then this form takes me 25 minutes, because then I have to delete things, because as you can see, this 0 here, it becomes hard and I can't delete it if you take all my formula boxes away. Like right now I can delete it, but you can see I cannot delete this. I just attempted to, and you can't tell. So if this amount, or if these amounts were 0, but you really have an invoice of $123,000, then I have to start editing stuff away. And then if the scan is bad, I might actually have to ask for a new form. So I'm pretty sure all of you have already digitally signed, but I cannot stress enough how much I appreciate the digital signature because of how much time. I mean, I could do 3 or 4 of your guys' invoices within half an hour versus 1.5 per hour. So it saves you time, saves the taxpayers time, you get paid faster, and then I feel more productive. So are there any other questions anyone might have? Because that's all I got.
Well, thank you so much, Amy, for joining us and walking through the invoicing process. And, tips and tricks of what we can do to do the invoicing better and speed up the process.
Yeah, absolutely. My pleasure.
I'm not hearing any questions, but if anyone has a question later on, please don't hesitate to reach out to Michelle or I, or sending a question on to the beach monitoring email. If it's something that we can answer quickly, we will do so. If it's something where we need Amy to weigh in on, we'll loop her in. And I think, does anyone have any other questions for this meeting in general? Or if not, I'll happily end the meeting 20 minutes early and let you guys get back to your day and your work. Hearing none, I'll end the meeting. Keep your eye out for meeting sometime in spring. We'll announce it through our email subscription list. And thank you everyone for joining us today. Let us know if you need anything for the beach program. Thank you.